Hi everyone! Today we're going over Chapter 2 of Biochemistry for the MCAT, which talks about enzymes. Chapter 2.1 deals with the role of enzymes as biological catalysts. All enzymes are catalysts, and you can tell when you're dealing with an enzyme because they'll end with the ending ase. Here I've drawn a classic reaction diagram of an exothermic reaction, which means that the energy of your reactants is higher than the energy of your products. So this reaction should happen spontaneously or without an additional input of energy. However, as you can see, in order to get from your reactants to your products, it goes over this high energy state called the transition state, and the amount of energy you need to add to your reactants to reach this high energy state is called the activation energy. So what it means for something to be a catalyst is that it doesn't impact the amount of energy that your reactants have or your products. So it doesn't make this reaction any more favorable, and it doesn't increase the amount of product that it's possible to make. However, it lowers this activation energy, so it makes this reaction happen faster. So more molecules will have enough energy to complete the reaction from reactants to products. So enzymes impact the speed or the rate of a reaction and not the equilibrium constant. There are 6 common types of enzymes that the MCAT likes to test on. The first is called an oxidoreductase, and as the name suggests, an oxidoreductase catalyzes a redox reaction. So it moves an electron from one place to another, oxidizing something and reducing something else. Dehydrogenases, reductases, and oxidases are common examples of oxidoreductases. So when you see these words, know that a redox reaction is happening and electrons are being transferred. The second type of enzyme is called a transferase. Transferases transfer entire groups of atoms instead of just electrons. A common type of transferase is a kinase, which transfers a phosphate group. This is really important because ATP is the main carrier of energy in our bodies, and ATPs operate by removing and adding a high energy phosphate group. The third type of enzyme is called a hydrolase, and hydrolases undergo hydrolysis reactions. So a hydrolysis reaction is when you want to break something apart, and in order to do it, you must add a molecule of water. So this attaches an OH group to one side and a hydrogen to another side, breaking the molecule apart. Last lecture, we learned about peptide bonds, and when you form a peptide bond, you release a molecule of water, and in order to hydrolyze a peptide bond or break the 2 amino acids apart, you have to add an OH group to one side and a hydrogen to another side. Common hydrolases are phosphatases, peptidases, nucleases, and lipases. The fourth type of enzyme is a lyase. This is similar to a hydrolase in that it breaks bonds apart. However, you don't need a water molecule to make this happen. Something really important about lyases is that their reactions are reversible. In fact, the reactions of all enzymes are reversible, except the backwards energy is different from the forwards energy. So in this reaction, your forward reaction needs a smaller activation energy than your backwards reaction, which requires a larger input of energy. Some reactions in biological systems do look like this. Other ones have products and reactants that are more equal in energy. So in this case, an enzyme will make the backwards as well as the forwards reaction faster. And because these reactions are reversible, lyases can also operate in the backwards direction as synthases. So if you see a word synthase, you know that it is the backwards reaction of a lyase. The fourth type of enzyme is called an isomerase. And as the name suggests, isomerases make isomers, so they can make either stereoisomers or constitutional isomers. You'll see isomerases a lot in glucose metabolism because isomerases can isomerize one sugar into a different sugar. And so these isomerases move atoms as well as electrons around. However, at the end, you get a molecule that has the exact same molecular formula. The sixth type of enzyme is called a ligase. A ligase is similar to a lyase in that it does addition or synthesis, and it can also break things apart. However, ligases often act on much bigger molecules, so they require ATP, or they require an input of energy in order to make this happen. Chapter 2.2 is all about the mechanisms of enzyme activity. The molecules that an enzyme acts on are called the substrates. And in order for any reaction to happen, the enzyme and the substrate first have to come together and bind, and this forms what is called the enzyme-substrate complex. There are 2 theories about how this can happen. The first is called the lock and key theory, and I've drawn a picture of what this might look like here. So this means that the enzyme is perfectly shaped to fit the substrate. So because they're perfectly shaped to fit each other, when they find each other, they fit together perfectly like a lock and a key. The second theory is called the induced fit model, and this means that the enzyme is not perfectly shaped to fit the substrate. However, they're shaped similarly enough so that they can find each other, and when they bind, the enzyme changes shape and it becomes something that fits the substrate better. And the induced fit model is the model that is more accepted in biochemistry now because it has been shown that enzymes do change shape and undergo conformational changes when a substrate is bound. There are several things that help enzymes do their job. Two of these are called cofactors and coenzymes. Cofactors are inorganic, so cofactors are molecules like metals that help an enzyme do its job. Coenzymes, on the other hand, are similar to cofactors, however, they're organic, so vitamins are examples of coenzymes. Apoenzymes is a name you give to enzymes that don't have their cofactors or coenzymes, and when they are bound to their cofactor or coenzyme, you call these holoenzymes. Prosthetic groups are names you give to either coenzymes or cofactors when they're really tightly bound or when they're covalently bound. So if a cofactor or a coenzyme is permanently bound to your enzyme, you call it a prosthetic group, but if it comes in and out of your enzyme, then you wouldn't. Chapter 2.3 is about enzyme kinetics. This means the concentration of your substrate, and this means the concentration of your enzyme. So here I've graphed a plot of reaction velocity at constant enzyme concentration. So if you have constant enzyme concentration and you have no substrates, your reaction will not proceed at all. And once you begin to add substrate, your reaction will start to get faster very quickly because all of your enzymes are empty, so any little bit of substrate you add will find an empty enzyme immediately. So the reaction gets very fast, but as you keep adding more and more substrate, it becomes harder for a substrate to find an empty enzyme because most of the enzymes are now occupied by substrates. So your reaction still does get faster, but it does so more slowly until we reach this point called Vmax, which is the maximum velocity of your reaction. And this happens when all of your enzymes have a substrate. So if you increase your concentration of your substrate, you'll reach Vmax, and then your reaction cannot get any faster. So we have this point at half of Vmax called the Km. What this means is that this is the concentration of substrate that you need in order to get to half of Vmax. And this is important because this is different for every single enzyme. And this is also known as the substrate affinity, or how much your enzyme likes to bind a substrate. So if your Km is very low, that means that your enzyme will bind a substrate very, very fast. And if your Km is very high, that means, like, let's say if your Km was here, then that means that you need a lot of substrate in order to bind half the enzymes, which means that your enzyme is not very likely to bind a substrate. And by definition, the Km is the concentration of substrate at half the Vmax. Here I've written out the equation for an enzymatic reaction. First, you have your enzyme and your substrate, which become the enzyme-substrate complex, and once the enzyme does its job, you get the enzyme and the product. This rate constant here is called kcat, and kcat depends on how fast the enzyme can do its job on the substrate in order to make it the product. This is a generic chemical reaction. You have A plus B becomes C plus D. So if you wanted to write the rate of this reaction, you have K, which is the rate constant for this reaction, times the concentration of A and B. This does not depend on the concentrations of C and D, because the rate of a reaction depends only on how often A and B can find each other and undergo their reaction. So if we wanted to write Vmax for this enzymatic reaction, it would be something similar, where kcat is your rate constant, and we only multiply it by the concentration of your enzyme, because at Vmax, the concentration of your substrate is maximum. So Vmax does not depend on the concentration of your substrate. The michalis-menten equation is the equation we use to find the velocity at any concentration of the substrate. The michalis-menten equation is Vmax times the substrate divided by Km plus the concentration of your substrate. This makes sense because if we have no substrate, then you would have 0 as your velocity. And if you had the concentration of substrate that is exactly equal to half of Vmax, so if we substitute Km for S, then we have Km over 2Km times Vmax. So we would have 1 over 2 times Vmax, which is exactly what it should be at Km. And this is another way of writing the michalis-menten equation. This reaction is sometimes even more helpful, and how you can get this is by substituting Vmax for Vmax equals E times Kcat. Another useful way to display your data is with the lineweaver-burk plot, which is just the inverse plot of what we had up above. So instead of having your reaction velocity on the y-axis, you have 1 over reaction velocity. And instead of having the concentration of substrate on the x-axis, you have 1 over the concentration of your substrate. So when we plot it this way, we will get a linear line, and on your y-intercept, you will have 1 over Vmax. And here you can find your Vmax directly. So if you plotted your data this way, you can only find Vmax as something that your velocity will never reach. So as you add more and more substrate, your velocity will get closer and closer, but you'll never reach it. But here, if we plot the inverse, we can find exactly the number that Vmax is. And here on your x-intercept, you'll have negative 1 over Km. Some enzymes have a characteristic called cooperativity. So your enzyme may have more than one active site, which means it has more than one location where it can bind a substrate to do its job. Binding one substrate may cause your enzyme to be more likely or less likely to bind a second substrate. In the case of hemoglobin, if your hemoglobin molecule binds one molecule of oxygen, this makes it more likely to bind a second. And if it binds a second, it makes it more likely to bind a third. So this is called positive cooperation. And positive cooperation will have a Hill coefficient, which is greater than 1. Your enzyme may also have negative cooperation, which means that if your enzyme binds one substrate, it can either physically block the second active site, or it can cause your enzyme to change conformation to make it less likely to bind a second substrate. And this will have a Hill coefficient that is less than 1. If your Hill coefficient is exactly 1, that means you have no cooperation. So if you have a Hill coefficient of 1 with no cooperation, your reaction velocity plot will look exactly like this. However, if you have cooperation, your plot will look kind of like this. This is called a sigmoidal plot, and you can see it kind of forms an S shape here. Chapter 2.4 is about the effects of local conditions on enzyme activity. As a side note, the words enzyme activity, enzyme velocity, and enzyme rate mean the exact same thing. They mean how fast your enzyme is acting on the substrate in order to create the product. The first factor is temperature. So the optimal temperature for enzymes in the human body to function at is at body temperature, which is 27 degrees Celsius. If you take the enzymes out of your body and you bring them into a lab, you'll find that their rate doubles every time you increase the temperature by 10 degrees Celsius until you reach body temperature. And then if you increase the temperature even more, your enzymes start to denature, which means they start to break apart, and then you will have no enzyme activity because your enzymes will no longer be functional. The second factor is pH. So enzymes function best at body temperature and body pH, which is 7.4. So if you lower the pH or if you raise the pH past the pH of your body, then your enzymes will no longer function as well. The last factor is salinity. And salinity, it kind of depends. So salinity doesn't have a high effect on enzyme activity. However, if you have a cup of salt water and you just throw an enzyme in there, your enzyme will denature. Chapter 2.5 is about the regulation of enzyme activity. There are two types of feedback regulation. One is called feed-forward regulation, and the other is called negative feedback. Here I've drawn a generic metabolic pathway. You have substance A, which needs enzyme A to become substance B, and so on until you reach your product, which is substance D. Feed-forward regulation is if, for example, you suddenly get a lot of substance A, and this enzyme B here is typically in its inactive form. So you need a way to tell enzyme B, hey, it's time to become active and make more of substance C. So your substance A can go forward and activate enzyme B, or your substance B can go forward and activate enzyme C, or your substance A can go and activate enzyme C as well. So feed-forward regulation is when a substance earlier on in the pathway goes forward to activate or deactivate an enzyme. Negative feedback is when, if for example, we have now made too much of substance D. Substance D can go back and inhibit enzyme C, B, or A. This is called negative feedback because it tells you that we've made too much of this, it's time to stop. There are different kinds of inhibition for enzymes. One is called reversible inhibition. Reversible inhibition means that this inhibitor can bind and it can unbind later if necessary. Competitive inhibitors are called competitive because they compete for your substrate in order to bind to the active site. If you have a high concentration of your competitive inhibitor, they will bind to the enzyme's active site and then the substrate is no longer able to enter. However, you can overcome this effect by adding more of your substrate, because if you add more of your substrate, then the enzyme will find the substrate first. And so if you look here at the lineweaver-burk plot, the black line is when you have no inhibitor. The Vmax with a competitive inhibitor is the same as with no inhibitor. This is because if you have some of your competitive inhibitor and you add a lot of your substrate, so if you add so much of your substrate that the concentration of your inhibitor is negligible, the enzyme will only find your substrate, and so the maximum velocity is still the same. However, the Km is now greater because your Km is the amount of substrate you need to be at half velocity, and you'll need to add even more of your substrate in order to get to 1/2 of Vmax than you would need to without your inhibitor. Another type of inhibition is called non-competitive inhibition. This type of inhibitor doesn't bind to the active site, but it binds to the allosteric site, which is a different site on your enzyme that when something binds to it, it causes a change in the shape of your enzyme that makes your active site no longer able to do its job. So non-competitive inhibitors cannot be overcome by adding even more of your substrate, because non-competitive inhibitors make it impossible for anything to enter the active site. If you look at the lineweaver-burk plot, a non-competitive inhibitor has the same Km, which means that you need to add the same amount of substrate in order to reach Km, which is half of your Vmax. This is because your Vmax is now lower than it was before. Your Vmax is lower than it was before because even if you add infinite amounts of substrate, it's no longer possible for every single enzyme to bind a substrate. But if you wanted to get to half of this value, half of this lower Vmax, you would need to add the same amount of substrate that you needed to before. Mixed inhibition is when your inhibitor has different affinity for your enzyme and your enzyme-substrate complex. This means that your mixed inhibitor could have very high affinity for your enzyme-substrate complex, which means that your inhibitor likes to bind to the enzyme-substrate complex, which makes the enzyme more able to do its job, which means that the inhibitor actually helps lock the substrate in and helps the enzyme bind. It can also have higher affinity for the enzyme, which means that it can act kind of as a non-competitive inhibitor. So this can increase or decrease your Km. Another category of inhibition is called irreversible inhibition. So with reversible inhibition, you can undo the effect by adding more substrate or by removing the inhibitor from the solution. However, with irreversible inhibition, the enzyme is turned into a deactivated form, which means that just removing the inhibitor will not turn the enzyme back into its activated form. Regulated enzymes can be regulated by several different things. Allosteric enzymes have an allosteric site, which is a site that's not the active site that molecules can also bind to. Allosteric activators are substances that if it binds to the allosteric site of an enzyme, it turns it from its deactivated form into its activated form. So this type of inhibitor is actually an activator, which causes an enzyme to do its job faster or better. Allosteric deactivators can bind to an enzyme and cause it to become deactivated, so the enzyme is no longer capable of doing its job. Other enzymes can be covalently modified. They can be modified by phosphorylation. So if you add a phosphate group to an enzyme, it can either do its job better or worse, depending on the enzyme. Glycosylation is also a way that you can covalently modify an enzyme to either become activated or deactivated. Zymogen is an inactive form of an enzyme. So some enzymes, such as digestive enzymes, are really dangerous and you don't necessarily want them to be active from when they're created. So a zymogen can be created and then delivered to its final destination and then become activated through covalently modified ways or through allosteric sites. Once again, thank you so much for watching and see you in the next episode.